So as you guys probably know, every major crop is threatened by pathogens. Billions of dollars are lost every year to pests, and tons of pesticides are sprayed. And I am from Idaho originally, which is similar to Holland in that we love potatoes. So here I have some pictures of potatoes. And this shows a variety that's susceptible to late blight and one that's resistant to late blight. And as you can see, late blight has the ability to decimate fields. And since crops are normally grown in monocultures, if one plant is susceptible, then all of them are. And in addition, pathogens are able to evolve and adapt rapidly and to overcome resistance. So breeders have to be constantly coming up with new varieties with novel genetic resistances. So I studied downy mildew of lettuce, and there's a picture of it. And it's a serious issue in both California and in Europe as well. Lettuce is a $2.9 billion crop in California, and downy mildew is the most damaging foliar pathogen. So over half of fungicides are applied specifically to target this pathogen, and there's no effective organic control currently available. So growers rely a lot on genetic resistance. Another challenge is that there's multiple downy mildew races present in California that are capable of sexual reproduction, which can result in new pathotypes and new ways to overcome resistance. So downy mildew is an obligate biotroph, which means that it can't live and reproduce without a living host. And this love story is one-sided because downy mildew extracts nutrients from the host, and while it also suppresses the host immune system. So this results in lower yields and increased risk of secondary infections, without the lettuce getting any benefits, of course. And this, of course, happens only in lettuce that cannot resist the pathogen. So how do pathogens manipulate their hosts? So the pathogen grows intracellularly here and forms little structures called hostorium. And this is the site of secretion for most effectors. There they go. And they enter the host cell and disrupt metabolism. They also suppress the immune system. And as a counter defense, of course, plants have evolved resistance proteins that recognize these effectors and trigger programmed cell death, which stops the spread of the pathogen. So this creates selective pressure for effector genes to evolve to evade recognition. And as this happens, the resistance proteins likewise evolve to continue to recognize effectors. So both pathogen and the host have to keep running and running just to stay in the same place, like the Red Queen in Alice in Wonderland. So since effectors are major pathogenicity proteins and they're also the determinants of plant resistance or susceptibility, there's a lot of interest in accurately predicting effectors. And in theory, this is really simple. You just take your pathogen, extract the DNA, sequence the genome, find the effectors, and you can screen germplasm for new R genes. In reality, the sequence data you get just looks like a bunch of A's and T's and G's, and it's not necessarily clear where the effectors are going to be. So so the question is, how do we find effectors in this alphabet soup here? And I'm going to talk about a couple different ways that have been used in the past and a couple different new ways that I've developed. So in oomycetes, such as Phytophthora and downy mildews, luckily we have a motif that is frequently associated with effectors and can be used to fish them out of the genome. So this was discovered back in 2005 by looking at alignments between effectors that were mostly cloned using map-based cloning. And just downstream of the signal peptide here, you have a motif called the RXLR, arginine, and the amino acid leucine and arginine. And then that's also associated with a downstream EER motif. And this was found in a number of Phytophthora effectors and then was used to find more effectors. However, it was not without its issues. The first one is that a lot of RXLR variants were found, especially in downy mildews. For example, GXLR, QXLR, RXLK, RBRN. And when you actually take all of the variants found in nature, as well as variants that have been shown to function in the lab, you end up with a really highly degenerate string of proteins. And this sequence is actually really likely to occur by chance because there's so many options. The false positive rate ends up being around 50%, It's a little bit lower if you combine it with EER, but not every RXLR has an EER, and not every EER has an RXLR. So it's not incredibly predictive due to the false positive rate. And there is a hidden Markov model for the RXLR, which is more accurate, but it doesn't seem to work as well outside of Phytophthora, because probably due to evolutionary divergence. So before I talk about our solution to the RXLR issues, I just want to show you the evolutionary relationship of the Phytoptheras and downy mildews. So this is the most recent phylogeny assembled by Kyle in my lab using a set of conserved genes in the oomycetes. And you can see that the downy mildews, these two groups here, are actually paraphyletic within the Phytotheras. And they also show a lot of divergence, probably due to careful co-adaptation with their hosts, because they're all very host-specific. They only grow on one host. So in this group of Phytotheras and downy mildews, it was recognized actually originally bioinformatically that a lot of them shared some similar C-terminal motifs. And then in 2011, 3 crystal structures of RXLR effectors came out, which identified this conserved fold, which was termed the WY domain because it had conserved tryptophan and tyrosine residues in it. So the structure is really similar between the different effectors that have it, although the amino acid sequence is actually quite diverse, as you can see. There are only a couple conserved residues, and the rest are really variable. And what was I going to say? I don't know. Okay, so basically, what my idea was to do is search for this motif and kind of ignore the RXLR for now. And part of the rationale was this motif's a little bit longer and maybe less prone to false positives. And also, this motif has been implicated in effector function through mutational studies, and it's also been implicated in our gene recognition. So in theory, perhaps proteins with the WY domain are more likely to be real effectors. And so what I did is I performed searches for this motif in lettuce downy mildew and a bunch of other oomycete genomes, Phytophthora and downy mildews. And I found that there were a lot of predicted WY effectors that didn't have RXLR, especially in downy mildews, but also in the Phytophtheras. So in purple here, I have RXLR with NER with WY. And you can see Phytophtheras have quite a few of those, less so in these downy mildews, and even less so in this clade of downy mildews. And then downy mildews in particular seem to be dominated by proteins with EER and WY, but no RXLR. And there also are quite a few WYs that don't have either. That's in blue here. And then for a comparison, I have RXLR ER with no WY, which you can see makes up a significant amount of proteins in Phytophthora and less so in the downy mildews. So basically, the proteins in yellow and blue here are proteins that would have been missed if you only searched for the RXLR motif in your genome.
Can I quickly ask a question?
Yeah.
This was trained on the Phytophtheras or not?
Yeah, the WY-HMM that I used was trained on Phytophthora, Infestin, Soji, and Remorum, about 700 sequences.
So maybe this is why you see so many of them in the Phytophtheras, not in the downy mildew.
Yeah, perhaps. And I mean, I built a model based on downy mildew WYs. It didn't predict any additional ones outside of the Phytophthora model, but maybe it could be improved in the future with more alignments or maybe some more crystal structures. Okay, so the first things I wanted to look at was to validate these effectors bioinformatically. So I looked for lineage specificity. So due to the rapid coevolution of effectors with their hosts, they're often species or genus specific. So and then that means that no homologous sequence can be found in closely related genomes. So I took the set of secreted lettuce downy mildew effectors and I used BLAST to identify homologs in pretty much every sequenced downy mildew and Phytophthora. And I also blasted it to another isolate of downy mildew just for a comparison. And you can see all of the proteins were recovered in that downy mildew, in the lettuce downy mildew. But then when you go to other downy mildew species, the best hits are all well below 50%. Same with Phytophthora. So, and they mostly go around 25% identity. So the Brevia WY effectors cannot be found in any of these other genomes, kind of suggesting that maybe they have co-evolved with their hosts. Another interesting thing we noticed bioinformatically was that even though some of these WY effectors don't have an RX-alar motif, they share one similarity in their N-terminal region. So after the signal peptide, which is highly ordered, you see an increase in disorder, which encompasses the region where the RX-alar motif either is or would be. And then for the WY proteins, which are in black and yellow here, you see an ordered region which coincides with the first WY domain. So right now this is an observation that currently lacks an explanation, but I suspect that it may be related to the mechanism of translocation of RX-alars that might be important in that process, so we'll see what happens in the future. But you can see that RX-alars and WYs do share that characteristic in the N-terminus. Okay, so next we wanted to see whether any of the effectors could be recognized by resistant varieties of lettuce through the action of their resistance proteins. And luckily there's a very straightforward assay to screen for this, and it works especially well in lettuce. And you use agrobacterium to express effectors transiently by injecting them with a needleless syringe. And if an effector is recognized, the cells undergo cell death. So it's a really obvious phenotype, and it indicates the immune recognition. So I always include several controls, a negative control, GFB, and 2 bacterial effectors that are known to cause cell death. And then on the other side of the leaf, I add my effectors. And you can usually do like 5 or 6 per leaf. The assay can be a little bit variable depending on the age of the leaf and the plant, so I always include the controls on the same leaf and only score ones where the controls work. Okay, so we tested 200 different lettuce varieties against all 21 effectors, and we did 4 replicates for each variety at least. And in all, that results in 37,000 infiltrations. So, and these were done, about half of these were actually done at Enzazaden in the Netherlands, and the other half were done at UC Davis by myself and a team of undergraduates. And obviously I can't show the reactions from all 37,000 on one slide, because it wouldn't fit. You can actually find them on the Mitchell-Moore Lab website if you're interested, but most of them basically were negative. Here I've shown all the positive results, which is quite a few. I have a number of different lettuce accessions along this way, and then I have my 21 different effectors. These 2 in black have RX-alars, but the rest do not. And you can see that there are a few effectors that are recognized by many different accessions of lettuce. And then there are also a few accessions of lettuce that recognize many effectors. And then the necrosis here is indicated in magenta. So you can see these 2 varieties recognize 5 different effectors, for example, and potentially a 6th one. And let's see. So we're following some of these up in the lab, trying to map out the genetic basis for this resistance. And we have some promising results. One of the most interesting set of effector recognitions was in this line called VIAE, and that has an introgression from the wild lettuce species Elverosa. And this genotype recognizes 5 different downy mildew effectors, and 2 of these effectors here are paralogs of each other, but the rest share no sequence similarity except for the fact that they have WY domains. So in the lab, we happen to have a pre-existing set of recombinant inbred lines between the VAE and a susceptible line that does not recognize the effector. So we were able to map resistance, and we found that it was completely linked to bremia, to downy mildew susceptibility or resistance. So all the susceptible rills did not recognize the effectors, and all the resistant rills did recognize the effectors. And all 5 effector recognitions were linked, suggesting that we have kind of a natural gene stack here, or potentially there may be one or two R genes that control recognition of multiple effectors. That's still being determined and mapped out, but we do have a number of candidate genes in the region here. Another line that we've done some genetic work is called Ninja, and this lettuce line recognizes BSW04P, which is one of the paralogs mentioned in the previous slide, but it maps to a different location in the genome. And we had an F2 population in the lab, so with a number of undergrads, I screened 200 F2s in one day, and Maria Trucco did the mapping and found a really strong peak on chromosome 4, and this is also co-segregating with DM4, DM7, and DM11, which were previously mapped in lettuce. And We also tested additional 3 genotypes of lettuce that had DM11, and they all recognized BSW4P. So it's a candidate AVR11 gene. My colleague Anna Geisvers actually has been following up on this, testing candidate genes, and got a result last week that was very promising, that we actually found the R gene. So I don't want to speak too soon, but yeah, it looks promising. The results will be out soon. So the main conclusion from the WY study is that there are functional effectors in downy mildews that have WY motifs and that don't have RXLR motifs. So if you only look for the RXLR, you're going to miss out on a lot of real effectors. And all these results are actually published on bioRxiv right now. So if you want to learn more, you can look that paper up. Also, the WY HMM is very easy to use in the program. The software is very nice. So if anybody wants to use it, I'm happy to send you the HMM and give you instructions for running it. I think another implication of this study for effector prediction in other organisms, from fungi to bacteria and other oomycete, is that we really need more effector structures, because this was how the WY domain was originally discovered. So I think having a more repertoire of effector structures would help us find more domains that we could search for and then find more effectors. And then this study got me thinking that one issue with even the WY domain guided search is that not all effectors have WY domains. We know not all RXLRs have WY domains and not all WYs have RXLRs. Motif-guided searches are really limited by what is already known. So it got me thinking again about the dark matter of pathogen genomes. And I was trying to come up with ways to find things that we don't know what they look like, basically. And I came up with 2 ideas for finding effectors that don't rely on motifs. And the first one is lineage specificity. And people have used this, I've seen it used several times in fungal genomes, but I hadn't really seen it applied to oomycetes yet. But basically the idea, as I talked about before, is because effectors are constantly evolving to evade recognition, they tend to be species-specific or genus-specific. And so you could use that to identify candidate effectors. So, and around this time, I got an email from Munir Nur, who's a very talented undergrad in computer science, and he wanted to try out his hand at bioinformatics. So I gave him this project, and he performed most of the analyses. And basically what he started doing is he took the lettuce downy mildew genome and looked for signal peptides to come up with the secretome. And then he blasted these sequences against an oomycete database, which consisted of all the sequenced oomycetes at the time. And then the hits which had an identity over 40% and over 30% coverage were considered conserved genes, which is really a pretty, this is kind of a loose metric for conserved genes, because 40% is not super high conservation, but we wanted to get the most lineage-specific genes possible. And we basically found around 2,600 lineage-specific genes, which is a little bit too many to start to do functional analysis. So we applied another technique to narrow it down a little bit. And that was, well, right now we're calling it effector O. And it's basically using machine learning on effector physiochemical properties. And the name is actually a nod to effector P, which was developed by Jana Sperschneider in Australia for looking for effectors from fungal genomes. So it basically, we trained a computer with a positive training set on a number of biochemical properties such as hydrophobicity, surface exposure, disorder, bulkiness, and interface propensity. And we also added N-terminal intrinsic disorder, because that was something we had noticed from the WIs. And we used a set of validated effectors from lettuce downy mildew, so all the effectors that I found that had a function. And then we also added a bunch of effectors from Phibase, which is the database of validated effectors. And it was about 100 proteins total, all from downy mildews and Phytophthora. And then the negative training set was a set of conserved secreted oomycete genes, with the idea that if they're conserved, they're probably not effectors. There might be a few in there, but in general, the data set will be mostly non-effectors. And we found that effectors and non-effectors have different biochemical characteristics. So you can distinguish them, or at least computers can predict whether or not it's likely to be an effector or not based on these characteristics. And so Munir tested a bunch of different algorithms for effector prediction based on these characteristics and found that the best one was this random forest algorithm. So this is like a rock curve, so it has a true positive rate versus the false positive rate. And as you can see, it's very accurate in predicting effectors. It has a low false positive and a good true positive rate overall. And I thought if you're not familiar with random forest classification, I thought I would just briefly go over it. Basically, you have a data set, and the algorithm makes a number of decision trees. So at each decision, it's like, okay, based on like the hydrophobicity, does this look like an effector or not? And then it moves on to the next characteristic and asks the yes or no question all the way down. And then it will build these trees starting with different characteristics and then take the grand average of all of the trees, which results in a pretty accurate prediction because it's like the strength of numbers, basically. So any error made by an individual tree is averaged out by the group of trees. And then, so when we combine the linear specificity and machine learning approaches, the overlapping set is around 683 proteins. And one good thing is that all of the secreted WIs are recovered by these two approaches, which we know are probably effectors. And this is also, it's still kind of a high number, but it's similar to the number of effectors that you see in Phytophthora genomes, so it's not extraordinary. And combined with genetic mapping data and population genomics, I think we're likely to find a lot of real effectors in this data set. So with that, I want to acknowledge everyone who's helped with this project, including all the current and former Mitchell Moore Lab members, our collaborators and funding sources. Follow us on Twitter. I run the Twitter. And I also want to give a special acknowledgement to all my undergrads, because without them, I wouldn't have been able to do so many projects simultaneously, and it would have taken me a lot longer to get all the results I've gotten. So I have 5 undergrads currently, and then I've had 10 who've helped me in the past with cloning and doing aggro infiltrations and such. So that is the end of my talk. I'm a little early, so I have plenty of time for questions. Now on to some lab results. So with help from several undergrads,